Imagine you are you are in a team of relay race and you are in the final lap, and then the intensity builds so much that the run has to be completed and they win something to win. That is how the competition to find out, to decipher the structure of DNA also has become in around 1950s, early 1950s. Until then, the composition is known, and there are also some things that are known, such as the helical nature of the DNA and some details of it. But to build the whole molecule, it's as if it is the building of the whole genome itself. To structurally we should see or describe the structure. I am Dr. BCMR, Assistant Professor at Shastra. So from the composition of the chemicals and the molecules or the subunits, the nucleotides themselves, there are several models that were proposed. One of it is by Pauling, and the Corey's model you call that. And Pauling is famous for deciphering the structure of proteins, at least the alpha helix. In his model of DNA, there were 3 strands, and the strands were put, the phosphate backbones were in the center, and the bases were on the outside, something like this. Of course, now we know that it's not correct. But why we are spending time on thinking on talking about this is the process of finding out or putting things together, and it's like solving a puzzle. And that is why how these people have studied is to learn how these people have studied or tried to solve this puzzle is important, because in the future when we come up with a question like this, how do we approach? It is about the approach that is being taken. So there were many other models. Some people came up with 4-strand model, and many were obsolete for us right now. But then thinking is a good thing. Then Watson and Crick built a model, and building a model gives a lot of it brings imagination in front of you, and then it can probably allow us to think more clear. And this is Watson, and here is Francis Crick, and he was about 33 years old, and Watson was about 23 years old at the time they figured out the structure of the DNA. They published their paper, the theoretical model they built, and in Nature in 1953, and this was published along with that of the back-to-back papers is along with Rosalind Franklin's the X-ray crystallography structures of DNA as well. I mean, the pictures of DNA. In their paper, they do acknowledge the contributions of Wilkins and the Franklin, because some of the unpublished experimental results and ideas were shared or were made available by Maurice Wilkins to Watson and Crick. As I said, this is the paper from by Rosalind Franklin. It is published just after the paper of Watson and Crick. So first thing, if a molecule has to serve as a DNA, as a genetic information, it is supposed to be irregular in sequence, which would mean that it shouldn't be just poly G like this. It should have some base, maybe A, G, G, A, C, T, A, some form of irregularity should have been there. Second thing is, it should be able to transfer information, that is inheritable information. One molecule giving rise to 2 identical molecules, as we discussed earlier. Because genetic information, the essential feature of genetic information is that it can give rise to, the information is inheritable. Something similar to that of one cell giving rise to 2 different cells, and they further probably can give rise to further cells. If the information to make these cells should have been there, these daughter cells should have had the DNA or the genetic information. The two daughter cells in this generation can have it if only it has been derived from the mother cell here. So the information should be inheritable. The information that Watson and Crick had was that it was clear that the chemistry was clear. There are 4 nitrogenous bases, A, T, G, C. It had a pentose, that is deoxyribose, and phosphates. And they know that the nucleotides, how they are. Putting together is something important. And they also had the information from Chargaff that the composition or the proportion of adenine is equal to that of thymine, and the proportion of guanine is equal to that of cytosine. And from Maurice Wilkins and Rosalind Franklin's X-ray data, they know that the structure is helical and it had repeating subunits, and there is also uniform in width. Now, whereas like Paulings, there are also other people like Fraser who also made something similar. Pauling's structure is that the phosphate backbone of the triple helix is inside, and towards the outside you had the bases, something like this. Whereas Fraser, another scientist of the same time, had a different idea, that he put the phosphates to the outside and the bases towards the inside. This is another idea that was given, and I think Fraser also had made hydrogen bonds between them. So these people had very good team of chemists, and they could discuss a lot of things, including the physiological state, the water, and then how these atoms would be in what kind of ionic state would they be, and what kind of interactions they can form, and the distances to form hydrogen bonds, and many other things. Say for example, like in Paulins, we have phosphate groups in the center, and that is not so possible because of the highly negative charges, and it is difficult to put them together. Instead, like in the Fraser's model, this is a very good model, except it's almost like Fraser got it, except there was one problem, is that he had 3 strands, and not that is one of the only flaw probably. Nevertheless, Watson and Crick built a 3D model. I think when we build these things and try to put our imagination and give a shape to our imagination, we can define the imagination and come up with some nice ideas like these. So they used some plates like these, cut them into shapes of adenine, thymine, guanine, and cytosine, and then they literally constructed the whole structure of the DNA. The phosphate backbone, or the DNA, this structure, the backbone is on the outside like this. This is another backbone, and the bases are put like in steps, one on top of the another. We will also discuss base stacking at a later stage. And then that is how they proved, and they published the results of this. So let's just have a quick discussion on the essential things when we discuss about it. The bases are in the center, and the phosphate backbone is in the outside. And here is the deoxyribose. These are deoxyriboses. And so here is a 5' phosphate group, and this would have been the 3' OH group. For the other strand, we would have had 5' phosphate here and 3' OH here. So if you see the orientation of these two strands, they are anti-parallel. One is going from 5' to 3', and the complementary strand is 5' to 3' in the reverse orientation. And that we also know Watson and Crick base pairing. They capitalized on the information from Chargaffs, and they have proven that, or they have modeled it such a way that guanine base pairs with cytosine with 3 hydrogen bonds, and adenine base pairs with thymine using 2 hydrogen bonds. And from Maurice Wilkins and Rosalind Franklin's X-ray crystallography structures, they have proposed that it twists and it is in a double helix, something like this. So they did not stop there, and once they figured all these things, they also write a nice piece in sentence in their article. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The best part of Watson-Crick model is, for me, is the base pairing. They have described these base pairings in great detail, and that base pairing is the key to determine what the sequence of the opposing strand would be. So if we had this, because there is a specific base pairing, the sequence of the opposite strand has to be, or the complementary strand has to be, something like this. And if we had anything like this and a strand or a double stranded DNA like this, the replication is very easy then. It would open up, and because, say for example, these two strands have separated like this, like we have discussed in while discussing about Chargaffs, we would have made the opposite strand that is this way. We now have exactly or identical sequence to that of this strand. So information is inherited by the daughter DNA molecules. It is an important thing that, say for example, we perform experiments, and sometimes not every experiment can be made, and in those cases we make where experiment cannot be made, we make a lot of observations or collect a lot of information that is available from other sources, and we build a model. I think as future scientists, the ability to make models don't have to be just the physical way of making these. It can be theoretical models in our with diagrams about how one event is influencing another event, and many other things. To build model is something we need a lot of imagination, and probably with these models we also call them as working model. We call it as working model because if it has not yet been proved or disproved, we keep it as something to guide further research or future research. And when techniques are available, and probably then we can test whether this model is right or wrong. It is something like the hypothesis. There are some things, I hope we learned some things about scientific thinking and the way it happens, and the structure of DNA, how it has come about. Thank you very much.